Okay, going through the chapter 11 questions of paper 3. Obviously, under normal circumstances, section B will be your option, but as we haven't yet learnt the option, that is the chapter 11 stuff. Okay, so describe how monoclonal antibodies are produced. I mean, this one may be easier just for you to look at the mark scheme, but we're looking for the fact that some kind of small mammal, usually a mouse, is injected with one type of an antigen. Then we remove the antibody producing cells from that animal. You take plasma cells that produce antibodies and tumour cells that divide endlessly, and you fuse the plasma cells with the tumour cells to make the hybridoma cells. And then you select the hybridoma cells which produce the appropriate antibody, and then they divide endlessly to produce the correct antibody. Okay, so question number 2, a nice fuzzy image for you to have a look at. I promise I didn't deliberately choose a fuzzy image, that is just the image that comes. And so it asks you, what is number 1? So that's this, something that's big and dark in a cell. It's usually the nucleus, and that's what this is. And then it says, identify the protein producing thick filament indicated by the dark band indicated by 2. So the thick filament, that is myosin. And then part 3 is to identify number 3. So that's this whole sort of section, which is going to be a muscle fibre. All right. So then for 3 marks, explain how calcium is involved in muscle contraction. So you could say that the action potential causes the release of calcium from the sarcoplasmic reticulum. It binds to the troponin and causes the tropomyosin to move, which then exposes the myosin binding sites. Obviously, that's more than 3 marks worth, but those are the kind of the main points that you need to be making. Okay, question 3. Outline the reasons for the differences in the blood concentrations between the renal artery and the renal vein. This is like a weirdly common question, or variations of this is like weirdly common, given that it's sort of a fairly, like, of course there's going to be a difference in the concentration between the renal artery and the renal vein. So the what are you looking for? So that the urea products are going to be lower in the vein due to excretion following the ultrafiltration. Oxygen is lower in the vein due to cell respiration. Carbon dioxide is higher in the vein due to cell respiration. Glucose is lower in the vein due to cell respiration. Sodium or iron concentrations will be different based on the production of urine. Or it could be lower due to the removal of excess. Drug or toxin concentrations will be lower in the vein due to its excretion in urine. So obviously you only need to make 4 points there, but to get the point you need to give the difference and a reason. Okay, so what is the difference and what is the reason for that difference? Then question 4, here we've got a picture of the kidney. So what is X? So X is the cortex, and then Y is the ureter. And it needs to be the ureter and not the urethra, because this is going from the kidney to the bladder, not through the body. Okay, and so then for B, distinguish between osmoregulators and osmoconformers. So in osmoregulators, the concentrations inside them remain constant, whereas in osmoconformers they're variable, which means that the internal solute concentration can differ from the external environment in osmoregulators, whereas in osmoconformers it's going to be the same, or vice versa. So birds, mammals, humans, freshwater fish are osmoregulators, whereas starfish, mussels, crabs, jellyfish, sea squirts, squid are osmoconformers. Okay, so again, just two of those points. Okay, question 5. Sorry, I possibly didn't give you quite enough space for some of these. I do apologise. So describe the process of spermatogenesis leading to the production of 4 sperm cells in a human male. So you say, where did it take place? In the seminiferous tubule. So you've got your diploid cells which grow, then they undergo 2 meiotic divisions. The primary spermatocyte carries out the first division, and then the secondary spermatocytes carry out the second division, and that produces the spermatids, which differentiate into the sperm cells and develop the tail, and the Sertoli cells help them to differentiate. Now, that's way more than 4 marks worth. So obviously you need to pick 4 marks from there. And then outline the roles of estrogen and progesterone in females during human reproduction. So they're there for the kind of thickening and development of and maintenance of the uterine lining. So then you've got estrogen will also stimulate the LH secretion, which stimulates ovulation. That progesterone maintains the uterus lining. That they both inhibit FSH and LH, and that progesterone is then helps to maintain the pregnancy by maintaining the uterine lining. That a fall in progesterone will lead to contractions in childbirth. You can talk about them both being there in the development of female reproductive organs and secondary sexual characteristics as well. Okay. And then question 6 for 3 marks. Describe the role of ADH. So remember, ADH stands for antidiuretic hormone. So it's released when the blood plasma water content is too low. So it makes the walls of the collecting duct more permeable to water, which then means that more water is reabsorbed from the filtrate. So more water is returned to the blood, and a smaller volume of more concentrated urine is excreted. Boom.